So what I wanted to share with you guys today is basically an overview on work that I have been doing with some wonderful collaborators on the anatomical mapping of particularly human subcortical function. Now, the basis of my research is always that I want to use images to understand eventually brain function. And how do I do that? I use images to derive maps. And it is to some extent very comparable to creating a map of the world. If you look at what you can see at Google Map, or you look at the outside of a brain, you see a lot of stuff. And you can zoom in, or you can make slices like I do. But this only becomes interesting once you can start deriving information from this. And you can do this by annotating maps, and you can indicate cities. You can map out the roads, and you can combine different types of maps using varying complexity as well as varying layers. Now, after you derive this information, you can start to understand, for instance, about traveling. So how long does it take to travel, for instance, from here to Paris? It should be, I don't know, maybe a 6-hour drive. And you can also start to understand where challenges in the road may actually be. So if suddenly your prediction also becomes that you will need 4 days to drive from Amsterdam to Paris, you know that something is missing in your maps. And that is the way I approach mapping the brain as well. Now, what we need for that is, first of all, images. And we started with this quite some time ago. And what we wanted to do is to create maps of the human subcortex that could cover the entire adult lifespan. So what we did is we decided already early on, well, we want to get the best anatomical information that we can get given the limitations that we have here in Amsterdam. So we collected a 7 Tesla submillimeter whole brain data set of 105 individuals. And for this, Matan Khan kindly developed a quantitative contrast in which we can derive quantitative maps from a single scan acquisition. And the big advantage of that is that all of these scans are intrinsically aligned. So you can use multiple imaging contrasts at the same time, and you can switch between them in order to find your anatomical structure of interest. And given that we are all very much for sharing data here in Amsterdam, we also made this freely available for further analysis for instance, you guys to use worldwide. Now, how do we go about that? What we try to do is not create one map, but to create an average map so that it can be used across individuals and across groups. And what we create is probabilistic maps, so averages. And what we started out to do is to do this by manual delineation. And here on the right, you see an example of how we did this with the red nucleus. The red nucleus is a very iron-rich structure in the subcortex and is therefore nice to visualize using QSM. Now, manual delineation, unfortunately, is extremely time-consuming, but it still represents the gold standard. So this is really where we started out. And we started out doing this for the globus pallidus interna, the external segment, the substantia nigra, the subthalamic nucleus, and the red nucleus. And we did this on 105 scans, and 2 people did this per structure, and also in both hemispheres. And what we created while we were doing this was also a standardized way to also do this. So we all use the same protocols. 2 individuals do the delineations then subsequently independently, and we do the calculations that follow on that. Now, what we ended up with then was a a set of maps of 5 structures based on 105 participants, and that represents already over 2,000 manual delineations. And these we could register to MNI so that other people could use it, and we shared those in the literature as well. But then, of course, we decided that that was not enough, and we wanted to increase on this. So we wanted to up the number of structures. And we still want to capture the inter-rater reliability. So we really do need these 2 individual operators to delineate these structures. However, we then did the calculation that we have more than 400 individual structures in the subcortex. And if we want to perform these on over 100 participants, it is not realistic to do this. We even made the calculation and we found out that we would need approximately 12,000 years of man hours for that. So we decided that that was not a very good idea, and we stopped it or reduced it at that time point. What we did do is we shared all the descriptions of the delineation procedures. And what we do now is we share those procedures, and we perform the delineations not on 100, but only 10 participants. We still keep in the inter-rater reliability, and we also shared some example data sets if you guys want to try this on your own data. Now, this was what came out then. We calculated dice coefficients, which is a measure of agreement between the raters. And we noticed that for some structures, it was relatively low. And for other structures, it was a lot higher, particularly for the larger structures. So what you do is you calculate the overlap, and you can imagine that structures that are very small or structures that have a lot of border voxels suffer more in their dice coefficient as compared to the to larger structures, because they have more center voxels. So what we decided to do then is we decided to also calculate a dilated dice coefficient, which is a little bit more forgiving on the border voxels and gives you a good indication on whether 2 raters still are looking at the same structure and delineating the same structure. And what you can see then is that actually also for the smaller structures and the structures with large numbers of border voxels, we actually still do quite well. So we are confident that this works, and we therefore continued with this as well. So the interim approach would be that the manual delineations are very important, but at the same time extremely time consuming. But they are a valid basis for confirming that automated approaches actually do their work as they're expected to do. And that was the next step. We have this wonderful collaboration with Pilou Bazin. And what Pilou did is he created an algorithmic approach. He developed a algorithm in which we feed in the manual delineations, which we create on 10 training sets, so 10 individual participants. And what MASP then gives back is an automated delineation of individual MRI scans that were obtained in the same way. And it automatically delineates all of these structures that we've been trying to delineate as well manually. So in our original release of MASP, 17 structures could be delineated automatically. These are all validated against these manual delineation approaches. And this also means that MASP can serve as a reliable alternative to this manual delineation, which of course is very nice since we're still collecting a lot of data worldwide. And I'm sure a lot of people would love to have their structures delineated automatically. Now, this MASP has the potential to being rolled out broadly, but I would always recommend to do proper validations on your data set. And for that, you can use the manual delineation protocols that we have shared in the literature as well. We had a lot of fun with this, and we continue to have a lot of fun with this. But unfortunately, we realized when we started this project already that we would not be able to delineate all of the smaller structures in the MRI, because we simply don't have the anatomical resolution to do this. Now, the same happened with the cartographers to some extent when they were exploring the world. And their solution to this problem was to draw in dragons, because that's what they could not see or could not map yet. There must the dragons be lying. Now, I don't think that I could convince any reviewer in the world with pictures of the brain saying, well, you know, I can't map that. That must be where the dragons lie. So we decided to go to a different approach. And since we can't get the detailed resolution that we need anatomically in vivo, we decided to move to a post-mortem approach. But the basis for the development of this approach was always that we wanted to create something that we could translate to the in vivo situation. So we basically wanted to cross the sticks and create a stone of Rosetta to translate between the two worlds. Now, we did this as following. We figured that the basis would have to be MRI. We needed MRI scans of these postmortem brains. And what we did is we wanted to create a system in which we had as little anatomical deformation as possible. Now, how we did that is we actually did not scan only brains, but we scanned the brain in the head. And then after the detailed MRI scanning, which was performed together with Nick Weisskop's group in Leipzig and Evgenia Kirilina, we performed the autopsy. We took out the brain, which we subsequently sliced, and we created microscopy stains covering the entire rostrocaudal axis of the brain. Every single slice we made, we also stained. Now, what we wanted to do is we wanted to reconstruct these slides into a 3D brain. And during the cutting process, we made additional pictures to facilitate this. And I'll show you in a minute how we did that. Then we create a 3D reconstruction, which we can co-align with the MRI, and at the same time, which we can analyze further. Here you see an example of, for instance, how we extracted the vasculature, as well as a method to image directionality of structures in the brain. So we wanted to keep the morphological structure as intact as possible. We also want to capture inter-individual variation, although that's extremely tricky in the postmortem brain, because the tissue is scarce and it is extremely tedious to process an entire brain. We really needed a 3D reconstruction, which we could co-register with MRI. So this is what we set out to do. And what I show you here is a 0.4 millimeter MPM of this brain. These brains went into the MRI scanner for about 16 hours. So they scanned them back and forth and front and top and whatnot. And I hope you can appreciate how crisp these images are, and that you can even start identifying the individual nuclei of the thalamus inside of these scans. So that was already a major step forward as compared to our database that we had for in vivo scanning. Now, we wanted to capture the inter-individual variation, and this inter-individual variation looks a little bit different as compared to when you study young, healthy in vivo brains. I put 3 examples here, and it's not really a fair comparison because here there are still some of the meninges, whereas these have already been taken off. But what you can see is that there's a difference in the size of the brain. There's a difference in the size of the sulci. This one is opening up already a little bit, or substantially actually. And this is something we normally see a lot in the postmortem brains. We have a strong bias towards elderly people, of course. Now, on top of that, once we do the autopsy, we can get small damages, or we can get slightly larger damages. And that is something that we simply have to accept. Now, what we developed then was an approach to process the tissue. And we had seen the beautiful work of Katrien Amunds et al. And on Big Brain. And we decided that we wanted to take a different route, because the creation of 7,000 sections and staining those all individually would mean that we would not be able to process more than one specimen within a reasonable time. So we wanted to combine histological and immunocytochemical approaches. So classic stains, but also stains for specific proteins. And we were hoping that we could create a combined myelo and cytoarchitectonic map. Now you can imagine that there's a lot of stuff that you can buy for this type of processing for rodents from biotech companies. We hope that we could also do this for our tissues, but this turned out to be really difficult. What we use has to be bigger than what is commercially available, and that works for basically everything. Even the microscopy slides, we have to have them cut specially. They are not standard sizes. And the same holds for stuff that we need when we're freezing the tissue for cutting, for instance. So these are just regular barbecue boxes that I get at our local grocery shop. And we place the formalin fixed and sugar protected brain inside, and we freeze the brain slowly. Now, for those of you working perhaps with rodents and have done cryo cutting, normally you use a little bit of tissue tack for that, and you can do maybe 40 brains with one bottle. We use 20 bottles for mounting a single brain, and it takes 3 days to properly freeze the brain. Now, what we do then is we travel with this brain to Utrecht, because they are the only ones in the Netherlands who have the setup that we would like to use. And what you see here is a cryomacrotome. So this is a machine that is set up to cut very large tissue specimens. And what we have mounted above it is a camera perpendicular to our specimen. What we have mounted is not only a camera, but it is a camera with a fixed focus. And that means that if our tissue block sinks, which it does during the cutting process, we actually have to adjust the position of the camera. So this camera is actually mounted on a spring that we can adjust very tightly. And every 5 sections, we have to manually rotate this and bring down the camera with 1 centimeter. Luckily, we have an awesome technician who helps with this. So I will show you now what this setup looks like, what it takes to create a section, and what one of these sections actually looks like once we've cut it. So we create the block face for reconstruction. What we do here is we take a picture of the block we're cutting. We turn on the machine and the machine starts to slide. I'm filming and catching the section, so it's not really sharp. You see that the section comes off. And this is a very sharp knife. You could lose a finger there. And this is what a section looks once we've captured it. And we put it in a little urine jar in some solution that actually protects it against further freezing damage. So this is the way the section looks when we've just cut it. And you can see there's quite substantial tissue deformation going on here. What we do is during the cutting process, every single time we have sliced a section, the machine goes back to a steady position, which allows us to take a photo. And this photo is made exactly in the same position every single time. And these we use to create these stop motion movies. So I'm just gonna let this run through so I can explain some things. What you see here is the ventricles. They are not filled with tissue tack because we simply can't get that in. If you're used to working with rodents, you will see something unexpected. There's still quite some blood actually left in the brain. And this is because we largely work with immersion fixation, but this is perfusion fixation. But the fixation procedure is never as efficient as in a rodent, because a donor has already been dead for a little while before we can actually perfuse the tissue. And the vasculature may not always be as nicely intact to rinse out all the blood. So this is not the way you would expect this to look exactly in the rodents, but it is what we have to work with, and it is one of the limitations of processing human postmortem tissues. Now we've run through the entire brain. I hope you guys saw the thalamus as well. If you looked carefully, you have also seen the locus ceruleus coming by. And we're soon gonna reach the level at which my technician and I always cheer and we go for a coffee. So that is at the end of the brain. And I can tell you that this movie takes 2 minutes to show, but cutting a brain takes roughly 2 days. And that is without the prepping. So what we end up with then is all of these nice block face images, and they are restacked. They are not registered together, they are restacked only. And what we can derive then already is these really nice surface reconstructions. But not only the surface reconstructions, the block face images already give you a lot of anatomical contrast as well. What you see here is that the ventricles are still largely filled with sugar water. So that's the cryoprotectant solution that we use. And what you also see is that this is the cutting plane. And these are actually virtual sections. These have been reconstructed. And what you see is that there's one small dark line here. We could have fixed that with contrast settings, but I just really like it to point out how thin the sections are. And what happened is that I actually leaned into the picture a little bit, and this is a shadow I cast. So what we end up with is 800 to 900 sections per specimen. We lose the anatomical orientation, which we have to regain later based on the block face images. What we also lose is small parts of the cortex and the cerebellum, because they are not attached to the brain and they just float off when we process the tissue further. The same holds for blood vessels. But we collect all of these sections and they can be stained after mounting or free floating, whichever we prefer. We can use all of them for microscopy. We create microscopy and then we scan the images on a flatbed scanner. And this happens at what I call a lower resolution, which is 21 micrometer in plane. And the reason why we don't go up in resolution is because a TIFF image is limited to 4 gigabytes. And we simply cannot create bigger images because the system won't store them. This is a file size that we can actually handle. So what we decided to stain for was myelin or axons to identify the myelo architecture, Nissl for the cyto architecture. We also created some sections for hematoxylin and eosin, and that is to determine that we really were dealing with control brains and not with brains with substantial neuropathology. And then we decided to stain for calcium binding proteins, which give us a lot more anatomical detail. But of course, we could have chosen from a wide variety of available antibodies to perform staining. Now, what we then had to do is we had to match the corresponding block face and the microscopy image. And here you see 2 corresponding sections. This is the same level, and this is the same level as well. And here you see an example of a small piece of cortex that has gone missing. So that's not in the reconstruction. Here you see the hematoxylin and eosin staining, and this is parvalbumin. Now, we created all of these microscopy sections, we digitized them, and then we really needed Pilou again. And what Pilou did was he first created an optimized 2D registration. So by creating block face images together with the microscopy sections, what you actually do is you turn a 2D to 3D registration problem into a 2D to 2D registration problem, which is a lot easier. And this is exactly what Pilou did. He registered the microscopy section to the corresponding block face. And he did that for all individual sections. And he ended up showing me this beautiful reconstruction. And I cheered. And then Pilou did what Pilou always does. And he said, nah, I think I can do better. And he went back to his drawing board. And what he actually included was a forward-backward co-registration on top of the 2D registration. And you can see that he was right. He substantially improved the reconstruction again to this beautiful image. Now, what we then ended up with is a combination of quantitative MRI parameters with, in the same space, block face, Nissl, silver stains for fibers. For the aficionados, this is a Bielszowski staining, and immunocytochemistry for calbindin, calretinin, and parvalbumin. So this is the cutting plane, and this is the reconstructed plane. Here we show you some examples of the microscopy sections, the way we digitize them, because you can appreciate the anatomical detail that these images actually give. And all of these sections we have stored. And if we want to go back for some reason to acquire higher power images, that is also possible. So if in the future systems become available that allow us to do this in even more detail, we can always go back. We don't have to process an entire brain again. So what we did is we basically reconstructed an entire human brain in 3D using microscopy and MRI. And what we can now do is we can go through it back and forth in all directions, and we can use this for mapping the brain in more detail. And particularly, you can imagine how useful the immunohistochemical stains can be for that. So we did all of this, and we also showed that you can use this information to use your favorite technique. For instance, if you want to study cortex, these reconstructions allow the inflation of the cortical surface and create cortical surface maps. All of that stuff works. And it also shows you the potential of the anatomical detail. Here, Pilou had a first go at extracting all the information that is in there on the vasculature. Now, interestingly enough, we did all of this work. We sent it into a journal, and the journal said, well, you know, this is only one brain. So we went back to the drawing table and we did it again, and we created a second reconstruction of a second brain. So what are we going to do with this information now? We want to extend the MASP algorithm that we created with more anatomical detail from these post-mortem data. And there is a challenge there because we have very little information on inter-individual variation. We only have 2 brains reconstructed for this right now. But still, it is more than nothing. And you could imagine that you can focus on a lot of subcortical structures, and a focus on the individual nuclei of the thalamus would be extremely interesting, and also a very logical step to take, of course. Now, as said, the anatomical inter-individual detail will be very difficult to capture. And the question then rises, how should we go about this? And I would really like your input on this. So we did a little digging in the literature, and luckily Jürgen May and Majtanic, they also went digging in the literature, and they made this nice overview of the atlases that are available on the thalamus. And what strikes immediately is that different authors have different names for different structures. Now, this is something we, of course, all know within the field of neuroanatomy. But there's an extra challenge in here. Is this can this then be ascribed to inter-individual variations, that these brains just look different? Or is it the inter-individual variation between the authors? Do they just perceive things different? So I'm not quite sure how to deal with that right now, because we also only have two brains, and it will be very difficult to identify the same structures in exactly the same way in both of these brains. Luckily, we do have the immunocytochemical markers, which severely help in identifying individual thalamic nuclei. And this is the type of images that we would then be looking at. So what I created here is kind of a pseudo-fluorescent image in which we combine the parvalbumin, the calretinine, as well as the calbindin staining. And you can see all of these individual nuclei of the thalamus that suddenly start to light up. And we can use the same approaches that we have been using for delineating these individual nuclei, and we can create 3D reconstructions of these. And we will do that. It's just, it's a lot of work. So the future outlook of the thalamus would then be that we would like to extend MASP with mark structures in vivo, because also in vivo we seem to still be able to push the anatomical resolution a little bit further, so that we at least can get some of the larger structures in the thalamus. We then will supplement it further with the postmortem data. And something else that we would love to do is to extend the postmortem pipeline to also investigate what happens to cortical as well as subcortical structures in neuropathology. Because that is something we do encounter a lot, of course, in the postmortem tissues as well. Yeah, so I'm going a little bit faster than planned because I expected to be interrupted. That brings me to my conclusions. We created a 3D atlas of the human subcortex, which we feel benefits very much from a multimodal approach. And I think also that in the future, we would want to work by creating all the available data sets worldwide, creating an incremental atlas. All the data that we have created can be transferred relatively easy to MNI space, which allows the reuse of the anatomical data for fMRI data. So placing anatomical markers at the spots where you see your activation. In vivo, we can capture the inter-individual variation in healthy brains. And we can obtain a high number of observations, whereas post-mortem, we get all of the nice anatomical detail, but lower numbers of observations. So since we are strong advocates of open science, we share all of our data freely with the scientific community. And we do that by including digital object and identifiers in all of our publications. And those will link you to our data sets and our atlases if you want to reuse these. Yeah, so we hope that this atlasing process will improve the MRI atlases further, and it will hopefully also resolve controversies in MRI observations, because we have been working also with the group in Leipzig on projects identifying the nigrosome. And the nigrosome described in MRI does not seem to match exactly with the nigrosome as we know it in the field of histology. And I know there are more examples where the anatomists working on MRI identify structures slightly differently as compared to those who work on histology. To give you a peek into our future outlook, I would like to share this with you guys. So this pipeline is relatively flexible, and what you can do, because you have to process the brains sequentially, you can actually adapt the histological staining protocols towards whatever brain you have under your hands. And one of the brains that we hope to study as a non-demented control actually turned out to have substantial neuropathological alterations of the Alzheimer type. So what we decided to do then is, instead to stain for the calcium binding proteins, we changed the protocol and we stained for the amyloid and tau proteins, the proteins that accumulate in Alzheimer's disease. And what we found now is that we can also create a 3D reconstruction of the distribution of these proteins. And what we found very striking is that the distribution actually is not it's not symmetrical. So what you see here in blue is a Nissl stain. In red, you see the distribution of the amyloid beta protein. There are still few artifacts left in here. And what you will see pop up in green soon is the hyperphosphorylated tau, which is the other protein that accumulates in Alzheimer's disease. You can see that the amyloid stays nicely on the cortex, whereas the tau here also in the cortex is limited to very specific spots, and also only on one side of the brain in this specific area. Now, what you will see soon is that the entorhinal cortex and the hippocampus also show substantial accumulation of the hyperphosphorylated tau. So this is what you're starting to see here. You can also see that the tau stays at the base of the brain. It does not extend in the same directions as the amyloid. And here you can see the head of the hippocampus coming in on one side, and you can see the hippocampus on the other side. And we thought this was a brain with a non-demented control. It was a non-demented control clinically, but when we looked at the hematoxylin and eosin stains, we found out that there were substantial plaques. So we decided to stain like this instead and perform a more detailed neuropathological diagnosis. And we found out that this was actually a patient who was at Braak stage 5. So this is not beginning Alzheimer. This really is full-blown Alzheimer pathology, which did not match the clinical observations at all. So that is another challenge that we are facing, that the clinical observations do not necessarily reflect the same thing as the microscopy. So with that, I think this is the most impressive slide that is actually in the entire presentation. This has been a tremendous amount of work so far, and it could not have been done without the driving force of Birte, the excellent technical skills of Raveen, and of course, all the wonderful work that Pilou does on all these reconstructions. So with that, I'm happy to take all of your questions.